Okay, well, thanks very much for asking me to talk about some historical diseases of trees. These are caused by species of Pseudomonas. So it's going to be a very short summary. I'll try and introduce some new work as well. So the two diseases I'm going to be talking about are bacterial cankers of ash and cherry, or other Prunus as well. Now, both of these are widespread in Europe and in fact worldwide, particularly for cherry canker. Now, although they're both called cankers, they cause rather different symptoms, or very different symptoms. Ash canker is recognized by the dark lesions on ash stems, on ash wood, and these are typically surrounded by overgrowths from the plant tissue. It's almost like a gall, and some people suggested it ought to be called ash gall. So compare that with the cherry canker, and this produces a much more rapidly developing canker on woody tissue, dark brown lesions, and often they're associated with bacterial ooze and this rather disgusting gummosis symptom, which, where's my little thing here, is gummosis, which is a mixture of plant products. So both called cankers, but quite different diseases. The ash canker develops much more slowly. Hello, I'm not going forward. Might just be slow. Why is it not moving?
Can you do, I think where you were there at the bottom.
Okay,
there you go.
It. Yeah, I think it's just slow. So getting back to ash canker, what is it caused by? This was, the cause of ash canker was identified by Nellie Brown working in the States back in 1932. There's an excellent paper by her about this. And it's caused by Pseudomonas sevastanii, which is related to the olive knot pathogen. Well, sevastanii pathovar fraxini, which is related to Pseudomonas sevastanii, the well-known olive knot pathogen. And it's very interesting because she describes finding this canker in European ash trees, trees which had been introduced from Europe. And so maybe it's a good example, an early example of an introduced disease on planting stock. Now, in her paper, there's some very interesting observations that she describes the slow way in which the disease develops, although it can obviously end up killing trees. And her inoculation experiments to prove the cause of canker took her at least 18 months. It's in pictures after 18 months incubation. So slow disease confined to ash and just one bacterium causing it. You compare that with cherry and Prunus canker. Here you have multiple pathogens all related within the Pseudomonas syringae complex. And again, we go back almost over 100 years, pioneering work of Harry Wormwald at East Malling in the 20s. So it is 100 years since this really got going in the UK. And then Cross, also working at East Malling, identified 3 species of bacteria causing the disease, and that's Pseudomonas syringae, Pathovars morspinorum, race 1 and race 2. We now know that these are really quite distinct pathovars and shouldn't be considered races of the same strain of the bacterium. Anyway, our recent research, particularly in Europe, has found that canker-like symptoms are caused by other pathovars of syringae, Persicae on peach, Avi on bird cherry, and Cerasii on bird and sour cherry. Now, a lot of work has gone on with genomics of this, looking at sequencing the bacterial genomes, studying genetic analysis. And it shows that although they are quite different pathovars and quite distinct in many ways, they do contain a common core of inherited virulence factors. And in particular, they seem to have a suite of what are called effector proteins, which are injected by the bacterium into plant cells to suppress plant defenses and also to modify plants' metabolism to favor bacterial growth. So there might be suggestion here that the bacteria have evolved by gathering together these virulence factors and allows them to attack cherry. The challenge, of course, with a disease caused by multiple pathogens is trying to prevent it, because the resistance method or an approach to control that operates against one may well not work against all the other pathovars. So it makes it tricky. If we look at the life cycle of cherry canker, And this is pretty well established now, and it's established the work of Cross at East Morning again has provide the basis for what we know now. And if we start off with a cherry canker in developing in early spring, early summer, in spring and summer, early summer, you get the ooze and bacterial, high concentrations of bacteria within the canker. Rain splash spreads these onto the leaves and onto the rest of the shoot surface as well. So in the phyllosphere. And this is an important phase of development of the disease, the fact that it grows and can multiply on the leaf surface, where it interacts with other bacteria. So it could be an area where there's a lot of gene exchange, horizontal gene exchange between bacteria, and maybe lead to new forms of bacteria evolving. But of course, you can get symptoms in leaves, shot hole symptoms, and necrosis on the cherry fruit. And then various mechanisms, rain splash gets the bacteria onto the woody tissue where they enter into the wood, causing cankers through leaf scars or wounds. This diagram here doesn't show bud infection, and whoops, gone forwards now. And bud and leaf and flower buds can both be colonized by the bacterium as part of the xylosphere, and sometimes you can get blossom blast when the blossom just gets wiped out. Now, the bud infection has been studied recently by Maria Moroni Barder in New Zealand. There's quite a detailed study on that, and it's quite interesting it highlights the potential role for buds as a source of the bacterium to infect woody tissue. These are some results from the Maria Barder's work in Lincoln in New Zealand, and it's interesting because it's looking at the presence of Pseudomonas syringae pathovar syringae and Pseudomonas syringae mors brunorum race 1 in buds before bud burst and actually throughout the season. But it also shows the effect of copper sprays before bud burst. You can only really spray before bud burst because the copper sprays are sometimes toxic. So you can see here that high percentage of the buds, whether they were showing any symptoms or not, had the Pseudomonas within them. And the two were present within these buds at this site. Interestingly, Pseudomonas syringae mors brunorum race 2 is not found in New Zealand. So that one has been kept out for some reason so far. But it does show how the copper spray is really quite effective in preventing the colonization of the buds. And this is a bit back to front. It's New Zealand dates on the bottom, and I've just put summer so we know where we are. So the copper spray does work as a means of control to some extent. But of course, we really may need other means of control. You could get resistance to copper developing in the bacteria, and copper may be phased out as a treatment anyway. So what I like to focus on is breeding resistance. And if we first consider ash canker, with all the work going on ash dieback at the moment, the fungal disease, and looking for resistance there, sources of resistance, it would be good if there could be some screening linked to that, with all these accessions which are being gathered linked to bacterial canker resistance. But it may well be that we need to develop rapid pathogenicity tests on ash. This is something which is very much needed. We don't know much about the epidemiology of the ash canker pathogen either. So in cherry canker, there is some resistance available already. Well, perhaps shouldn't call it resistance. It's more like various reduced susceptibility. For example, the Merton glory, the cultivar, is considered less susceptible than something like Sweetheart or Napoleon. But there isn't any clear-cut complete resistance or immunity. But recently, work at East Morning by Michel Hulin, Andrea Badilio, and Francesca Cossu they've screened a vast number of wild cherries and Prunus species and found that there is resistance in both certain wild cherry and also in some Prunus species as well. And you can see the leaf test. This would be the ideal leaf test for susceptible here and resistant here. Doesn't always come out quite like that. But here's some real data on this with the scores, symptom scores. And I'd just like you to have a look at Prunus incisor, Fuji cherry. And this has been inoculated with various pathovars, avii, strains of syringae, strains of race 1, strains of race 2, and pretty much nothing, which is really good compared with the sweet cherry varieties here, which are go up to the maximum score. Pretty much. Whoops, done it again. Maximum score. Groton B, a wild cherry, is resistant to most strains as well. So there is resistance in the related species, and a cross between Incisor and Napoleon, the progeny from that have been studied, and they're turning out to be resistant as well. So there's hope that's an inherited, perhaps a dominantly inherited trait that might be useful for incorporation into sweet cherry cultivars in about 25 years' time. So the other approach, which is it's becoming quite topical, is the use of bacteriophages. But this, of course, has been around for a long time, the potential to use bacteriophages to kill pathogenic bacteria, whether human or plant. And that's a rather nice picture of a bacteriophage here, and it's attacking the bacteria. And this is what it does. There's definitely phages out there which kill Pseudomonas syringae, pathovar syringae. This is work by Mojgan Rabi at Birmingham. And here we have a pathogenicity test. You've got the bacteria growing in medium, multiplying with optical density. If they have the phage in there, it kills the bacteria, they get lysed. So this is an optical density measurement, and it goes right down. So they're not only killed, they're lysed completely as the bacteriophage gets in. And when it comes out, it kills and breaks the cell open. So there's huge potential here, I would say, for bacteriophages to have a role. But the problem with not just bacteriophages, but other antagonistic microbes in on the leaf surface or on the shoot surface is how to get experiments from the lab into the field. So I'll just finish there. I've covered most points. So the future targets, I would think, are rapid and reliable path tests for ash canker, understanding the epiphytic phase, what's going on in there, in both for ash and on the cherry, the role for antagonistic microbes in the phylosphere control, and in terms of understanding mechanisms of pathogenicity, dissecting what I call the effector functions in woody tissue. Why these bacteria are able to invade wood, I'm not sure. They don't degrade the wood. They get in there and they multiply within the living tissue within the wood. And of course, the incorporation of durable resistance into our trees. This cherry canker project is supported within the bacterial plant disease scheme, predicting the emergence of host-adapted bacterial phytopathogens. And the lead PI there is Richard Harrison, NIAB.